Thank you. It's very nice of you guys to stay. We've had a lot of good talks today, and some of what I'll have to say overlaps with some of the things you've heard earlier today. But what I really wanted to focus on is just sort of some practical things that I've learned as we've been doing some of these things. And you've heard about newborn screening, you've heard about exome sequencing, and I'll talk about each of those, but about maybe some practical things that we've learned that might be helpful as you do this as well. So I sort of subtitled this talk how we can catch things before the trouble starts, as in this fellow who's about to go overboard. So here's there's my disclosures. None of them actually are relevant to this talk, however. So I'm going to use the term, I'm going to be talking about early diagnosis of genetic immunodeficiencies, and I'm going to use the term PIDD for primary immunodeficiency disease, okay? So when you see that come up, and I'm going to talk about these topics. So the status of genetics and PIDD, sort of where are we at? You've heard about this a little bit earlier today. Then how we identify genetic defects in PIDD. You've heard a lot about that. And then I'm going to talk briefly about some surprises that have come up as we've been doing diagnostics genetically. We're going to talk a little bit about one of the challenges as we do diagnostics and doing genomics is how do you figure out which of those variants is the one that actually causes the disease? And I'll tell you about some of the things that we do in our lab to try and get at some of that. Talk about screening for PIDDs. You've heard about newborn screening. We'll talk a little bit at that, and then some speculation about where all of this is headed. Some interesting things that I think are out there. So if you take the whole pie of immunodeficiencies and you divide it up, this is from a publication actually more than a decade ago, but is probably pretty similar. This is in a large immunodeficiency practice, and it really, the distribution is about 50% of our patients have antibody deficiencies. These are going to be the CVID patients you've heard about, the patients with X-linked A gamma globulinemia. About a quarter of the patients have combined immunodeficiencies, so SCID or Wiskott-Aldrich syndrome. About 15% have neutrophil defects. About 10% have cellular immunodeficiencies. Those are ones that primarily affect just the T cell compartment. And then there's a little tiny sliver of the pie that is complement deficiencies. And this is pretty true from clinic to clinic and in terms of the breakdown. Now, you heard earlier today sort of the growth in the genetics, and I sort of chose to represent that this way. This is the STEAM textbook for immunology, and when version 1 came out, the first edition in 1973, the first the first molecular defect in immunodeficiencies had just been identified in 1972 by actually a biochemist at the University of Washington by the name of Elo Giblett. And Dr. Giblett found the absence of adenosine deaminase activity in a patient with SCID. And so it was the first link of a molecular defect to an immunodeficiency. And that was an enzymatic defect. She subsequently thereafter identified PNP deficiency, which is sort of similar. And then there was sort of a big lag before the genetic revolution in immunodeficiency came about. And that really came about in the early 90s and has exploded. As you can see by the 3rd edition, the 5th edition, the 6th edition was just published, but it's the only version I've seen so far is on the iPad, so it's much thinner than the 5th edition but would almost certainly be a couple of 5th edition size. It's really grown, and you heard that there are now about 270 molecularly defined disorders of the immune system, and so there's now about 21 genes that can cause a phenotype of severe combined immunodeficiency, 6 genes at least that can cause a hyper-IgM phenotype, and it goes on and on. So we'll talk about this a little bit later, but that's sort of the status of genetics in the immunodeficiency world. People who do this for a living, between us, we believe that in the end there will probably be about 500 single gene defects that cause immunodeficiencies. And so there's still, we think, a fair number to discover. And so the question is, how do we do this? You've heard a lot about ways that we sequence, and there's two major ways you've heard about that we sequence, by Sanger sequencing and by these next generation approaches. And I think the thing to remember here, and you've seen a figure like this earlier, is that really Sanger sequencing focuses on, Sanger sequencing is very involved. So it's very labor intensive, and what you do is you amplify each individual exon of the gene and then you sequence it. You amplify it by PCR and then you sequence it. And these new approaches that allow us then to step up to the exome or to the whole genome generate a lot of data. And I tell my patients that really what we're doing when we look for, when we're doing this is we're really looking for spelling errors, okay? And really what we're doing when we do whole exome and whole genome sequencing, I try to use this analogy because I think that this helps them to sort of grasp what we're trying to do with whole exome or whole genome sequencing. And if you happen to do this with your patients, this might be a useful analogy. So it turns out that the human genome is 3.2 billion base pairs, okay? And War and Peace by Leo Tolstoy, conveniently, many of you have probably read War and Peace. It's a large novel. War and Peace is approximately, it's just over 3 million characters, okay? So when you do whole genome sequencing, you are basically taking 1,000 copies of War and Peace, and you're sifting through them to find the one word that is misspelled or missing that causes their disease, okay? And that's what I tell my patients. And that's sort of, they get that. Now, the other thing I tell them is that the problem is that half of those copies of War and Peace are in UK English, where they spell color C-O-L-O-U-R, and half of them are in American English, where we spell color C-O-L-O-R, and you have to tell your computer algorithm to ignore those differences, but to focus in on the real differences. And it could be that there's a single letter that's out of place in those 1,000 copies, or it could be that there's one word missing, or maybe a whole sentence, or maybe a whole chapter is missing out of those 1,000 copies. But it's still a lot of work to find that, okay? Now, if you hone in just on the exome, as you've heard, the exome is about 1 to 2% of the genome. Now you whittle that down to just 10 to 20 copies of War and Peace, which is still a big task and still requires computer algorithms that are quite powerful to be able to do this. Now, as you can imagine, the chances that you're going to miss something sifting through 10 to 20 copies of War and Peace are relatively high, and they're completely dependent on how effective your computer algorithms are at analyzing that data. Okay, so as you've heard about, because of this emergence of these new technologies, it has offered this whole spectrum of options for doing genetic testing. You can hone in on a single gene. And so, as an example, in the immunodeficiency world, if you have a 5-year-old boy who has chronic cough, he's had a couple of bacterial pneumonias, you look at his lymphocytes and he has no B cells and he's got low immunoglobulins. He's a boy, no B cells, low immunoglobulins. Probably you're just going to sequence the BTK gene to look for XLA. You don't need to do whole genome sequencing in that case, right? However, if it's a girl who comes with the same story, well, there's actually many genes now that have been associated with congenital agammaglobulinemia or absence of antibodies, and in a girl it could be any of those autosomal recessive genes. And so then stepping up to a panel or to whole exome sequencing makes more sense, because it turns out that for most of the panels that are being offered, these next generation panels that sequence a lot of genes, for most of those panels, the cost is about equivalent to sequencing around 3 genes by the Sanger method. Okay, so if it looks like you're going to be going over 3 to 4 genes that you need to sequence, then it makes financial sense to step up to one of these panels, these next generation panels, or even potentially to whole exome sequencing. Okay, so and then of course you can ratchet that all the way up to whole genome sequencing. And each of these has pluses and minuses that we won't go through. You've heard a lot about that, but it's daunting as a clinician to know, where do I start? And I would encourage you, two words of advice, I guess. One of them is, if you have questions, don't hesitate to pick up the phone and call your favorite geneticist, or pick up the phone and call one of us who run labs. We're very happy to walk through the situation and try and help you to decide what would be the most efficient and effective way to do the testing on your patient. The other point that I would get to is that I always remind our fellows and our students as they're coming through, trust your gut. Trust your gut instinct. So if for instance, you had the patient that I just talked about, the 5-year-old boy with no B cells and low immunoglobulins, you sent the BTK gene and the results came back normal, you might say, oh, he can't have X-linked A gamma globulinemia because the gene was normal, okay? But your gut tells you he has X-linked A gamma globulinemia. He has to have X-linked A gamma globulinemia. He's a boy, he's young, he's got no B cells, he's got no antibodies. It has to be XLA. And we've had multiple situations in our diagnostic lab where we've done the sequencing by the Sanger method, and again, when you do that, you're just amplifying the exons and the immediate surrounding regions, and it turns out that the patient has a mutation deep in an intron that creates a new splice site and creates a new splice variant that doesn't make protein. And we've had many cases like that. And in those cases, what we do is, the clinical story, and your gut tells you it has to be XLA. And so that tells us we got to dig a little bit deeper. And so we sequence the messenger RNA or the cDNA instead of genomic DNA, and we get the answer that way. Or we use other functional approaches. So again, the point I would make, in this genomic era, the tendency is to order a genetic test, and if it comes back normal, to say, oh, they can't have that disease. And I would just remind you that your clinical instincts are better than genetic tests in many cases. And the genetic tests support your clinical instincts. And if you don't get the answer that you think you should get, sometimes you have to dig a little deeper, and sometimes that means you might have to go to a research lab to get some of those tests done. Now, what are some of the surprises that we've stumbled into as we've been in this genomic era? Well, one of them is that we are finding what I call fuzzy phenotypes, okay? So these, what I mean by that is that we're doing whole exome sequencing on patients who don't quite fit neatly in one of our diagnostic boxes. We do whole exome sequencing, and in our hands, and in the hands of many of the folks in the immunodeficiency world who are doing this, you do the exome sequencing, and it turns out that it's a mutation in a gene that's already been associated with immunodeficiency. It's not a new defect, it's already something you know. But it's a new phenotype of that disease. And so what whole exome sequencing is doing is it is expanding the phenotypic spectrum of many of our diseases. And this will be same across the board, whether you're doing immunodeficiencies or neurology or other things. I'm not sure what I meant by the next thing, when doing, but anyway, the other thing is that the next point that I would make is that it's important that we be able to do functional confirmation of the genetics. So you heard about all of these variants that you find. So it turns out that there have been studies done now looking at whole genome and whole exome sequencing just in normal individuals. So the average normal person has 20 loss of function mutations that are different than the ones that their parents have. Okay? We are all mutants. Okay? And so the point is that it's really important to have functional tests available, as you've heard, to then take these new unknown variants and test them and see whether or not those really do cause the disease. Boy, I don't know what I did here, but oh, you know what? This is the old version of the talk. That's why, I think. Let's do this. All right, here we go. I take that back. All right, a couple of slides in between there. All right, here we go. So here's the other surprises. So the causative gene is identified. So here's surprise number 2. The causative gene is identified in only 25% of cases, as you've heard about. So if you're sending sequencing, you're only going to find the answer in 25 to 30% of cases most of the time. one of the things that really just blew my mind, because when I was trained, I was always trained that you need to look for a unifying diagnosis. There's almost every patient has one diagnosis. Well, it turns out that genomics now tells us that's not actually true. Because in this study that I have down in the corner, which was cited earlier, which is the Baylor whole exome sequencing core that published their 250 whole exome, clinical whole exomes last October in the New England Journal, it turned out that 1 in 20, approximately, 1 in 20 patients has 2 causative mutations, either of which could have caused their disease. And this has now been borne out. I was just talking to Jordan Orange, who is who's the chief of immunology down at Baylor, and Jordan said that they've now sequenced 400 patients with immunodeficiencies, and this exactly, the exact number has borne out, that 1 in 20 have 2 loss of function mutations, both of which could cause an immunodeficiency, but together may cause a new unique phenotype that's a little bit rock and roll, a little bit, anyway, right? So it could be a little of both. And I think the first case of this that was published, as far as I'm aware, was a patient who had big deletions in the DOC8 gene, so had DOC8 deficiency, and big deletions in the CARD9 gene, so had CARD9 deficiency, and so had a somewhat unusual phenotype. So the other interesting things that came out of this study were that 83% of the autosomal dominant mutant alleles were de novo. And this gets back to the question that was raised earlier about newborn screening. Do you screen the newborns or do you screen their parents? Do you screen the carriers? And this is one of the reasons that we screen the newborns, is because 83% of de novo autosomal dominant mutant alleles were not present in either of the parents, and 40% of the X-linked variants also were not present in the mother. Okay, are new mutations. Okay, that hadn't shown up in that family before. So again, these are sort of the genetic surprises that I think have come out of this genomic era. So again, you generate a ton of data. You get all of these mutations, and you have to try and sort out which one is the causative mutation. And so we do that. We rely a lot on flow cytometry because, unlike a lot of other disorders, where you can't just take out the organ and test it and put it back, we have the luxury in immunology of being able to take out some of the cells, sample them, and study them, and we do that by flow cytometry. And for those of you who may or may not be comfortable with flow cytometry, I'll just give a brief 2-slide primer on what that is. So flow cytometers are basically fancy cell counters, okay? And what we do is we take a mixed population of cells, a mixed population of white blood cells, and we tag those with different antibodies that bind to markers on those cells that we want to study. And so one antibody, for instance, that binds to CD14 may be tagged with a blue fluorescent marker. An antibody that binds to CD19 on B cells may be tagged with a green fluorescent marker. And an antibody that binds to CD3 on T cells may be tagged with a red fluorescent marker. You throw those antibodies in, they bind to the cell that they're supposed to bind to. Then you take this whole mix and you put it into your fancy cell counter. And this is what happens. So the flow cytometer sucks up a single cell at a time, okay? And it sucks it up and it passes down this tube toward the detector. And as it passes through the detector, this single cell, a laser shines on that cell and causes whatever fluorescent markers are bound to the antibodies on that cell to fluoresce. And then there's an eyeball, and a little electronic eyeball, a detector, that basically says, oh, that cell glowed red, and my owner, the scientist, has told me that red equals T cell, so that's one T cell, and it drops it in the bucket, okay? And same thing for all the other cells, okay? So that's what this is flow cytometry. It's really just this simple. Now, what you get, though, is you get the ability to assay huge numbers of cells, so large populations that are statistically significant very quickly, okay? And so with that, we generate these flow plots that are dependent on how much fluorescence is on a particular cell. And so that's how the flow cytometers work. And they're very, very effective at allowing us to do a lot of functional testing. So what sorts of testing can you do with a flow cytometer? So one of the simple types of tests that you can do is, so say you find a mutation in, say, the BTK protein or the WAS protein. You find a mutation, it's never been reported before. You don't know whether it's going to be causative of the disease. Well, one of the simple things you can do is just use the flow cytometer to look, is the BTK protein or the WASP protein actually in the cells? Does the mutation just make it so the protein gets rapidly degraded, okay? And in fact, so we can do that just by looking at specific proteins. And here's some examples of some of the tests that we do in our lab, looking for Wiskott-Aldrich syndrome, BTK, both types of X-linked lymphoproliferative disease, FOXP3. So this is, as long as you have an antibody that detects the protein effectively, this is really possible with virtually any protein that you want to study. So just, is the protein present or absent? And that can get you a long ways. The second thing that we can do with a flow cytometer is that we can do immunophenotyping. So we can do very complex studies. You've already seen a little bit of this in Gubu's talk about how just by studying really the development of all of the B cells, for instance, that are floating around in the body, you can get some pretty good clues about where the genetic defect might be, because we know what genes, we know many of the genes that are required for a B cell to move from immature to mature, or mature to mature memory cells. We know a lot of those, and so if you find that they're blocked in that development, just by doing these sort of immunophenotyping studies, that gives you a pretty good idea of where the defect might be, and so that can be helpful. Thirdly, our functional assays, and this is really the simplest functional assay. You heard about this assay earlier. This is the assay that we use to detect chronic granulomatous disease. Basically, you take a dye that doesn't fluoresce, and when that dye is reduced, it fluoresces, and the dye becomes reduced when the neutrophils make an oxidative burst, okay? So basically, you incubate the neutrophils with this dye that doesn't fluoresce, you run them through the flow cytometer, and you see over to the left the dotted line. So this doesn't really work. Oh, it does kind of work. But to the left, the dotted line, that's the unstimulated neutrophils. They're very dim. It doesn't fluoresce. Then you stimulate them, and now they become very bright because they've made their oxidative burst, and that reduces the dye, it fluoresces, and the fluorescence shifts way over to the right. Now, in a patient with chronic granulomatous disease, it either doesn't shift at all in X-linked patients, or in an autosomal recessive patient like this one, it shifts a little bit. This is the easiest functional assay that we've got. But this one functional assay, which in our hospital, in our lab, the charge to the patient is like $86, okay? This one functional assay can diagnose X-linked chronic granulomatous disease or autosomal recessive chronic granulomatous disease, and you can do it in about 2 hours, okay? So that's the value of these functional tests. And again, there are now a whole bunch of antibody reagents and other reagents available that allow us to do other functional testing. And so here's a patient, for instance, with IL-10 receptor deficiency. And so what you're looking at here is you're looking at the ability of the IL-10 receptor to lead to phosphorylation of this downstream signaling molecule called STAT3. And so STAT3 binds to the receptors. It gets phosphorylated when IL-10 activates the receptor. And because we've got an antibody that binds only to the phosphorylated form of STAT3, you can see this big shift when you stimulate the cells with IL-10. And so if you look over here to the left, under the normal, red is unstimulated. Green is that there's now a lot of phosphorylated STAT3 because the fluorescence has gotten brighter. And as a control, we use IL-21. And in the patient who's got a receptor defect, we can immediately identify him because when we stimulate with IL-10, there's no shift in the fluorescence, okay? So there's no signal sent. So there's a whole bunch of these functional assays that we're now using. And really, our goal in the diagnostic lab that I run is that our goal is to really build out these functional assays to really test entire signaling cascades that are important for the immune system, so that when you get a defect, you identify genomics, you can very quickly do a functional assay to say the defect is in this pathway, and we've confirmed that that's where the defect is. So again, you really need these functional assays, and in order to make the diagnosis quickly, and when the kids are young, you have to be able to back up the genomics with functional tests, and you need to be able to do it fast because it makes a difference for therapy, okay? So let's talk a little bit about screening for immunodeficiencies, and I'll talk primarily about newborn screening for SCID, because that's where there's been a lot of work done. There's now a lot of experience on this. So just to remind you that January 21st of 2010, the Advisory Committee for Heritable Disorders voted unanimously to add SCID to newborn screening. So so you heard about how hard it is to get a test off the menu for newborn screening once it's on. It's probably it's not impossible, like getting it off, but it's vanishingly close to impossible to get one on. It's very difficult to get a test on the newborn screening panel, and there's this whole list of things that a disease these criteria that a disease has to meet to sort of get on the panel and to be recommended. And remember that as you heard, that newborn screening is an unfunded federal mandate. The feds say what you should test for, but it's up to the states to put this into practice. Okay, so there was a lot of work done to do newborn screening for SCID. One of the criteria for newborn screening is that the disease needs to be present in the population at least 1 in 100,000 births, generally, and it needs to have some sort of therapy, ideally. And in SCID, of course, that was easy because we've got a great therapy, which is curative. And so for us, this ended up being really a 5-year saga to get newborn screening approved and running in the state of Washington. And we just started screening in January, and I learned a lot of lessons in the process about working sort of working with Department of Health, which we've got an absolutely fabulous Department of Health in the state of Washington, a terrific newborn screening lab, but even with them all in and ready to start the screening as soon as we presented this to them 5 years ago, there were so many political hoops that had to be jumped through that it took us that long to get it to go live in our state. And that is one of the things I think that I learned, which I'll get back to in a little bit. This is the current map of newborn screening in 2014. The blue states are the states that currently have ongoing screening programs. Purples are the ones that have pilots that will be starting soon, or they'll be starting screening altogether, and the green states are the ones where there's not screening currently planned. And in fact, one of those states, Louisiana, screened for a while and then took it back off. So, breaking the rule, I guess, of getting it back off the menu. So anyway, but in that case, they made a decision to take it off. So, how is newborn screening for SCID done? It's pretty nifty. So, y'all may remember from your medical school or college immunology classes, and maybe not, that the receptor, the gene that encodes the receptor for the T cell receptor, or the gene that encodes the immunoglobulin molecule, it's made up of a whole bunch of segments, and that's how you generate the variability. So there's all these V segments, all these D segments, and all these J segments. And I think of them as sort of like beads on a string. There's a whole bunch of red beads, a whole bunch of blue beads, and a whole bunch of yellow beads, okay? And basically, in order to make a functional T cell receptor or a functional antibody molecule, you only need one V, one D, and one J, okay? So just randomly, the cell reaches over and grabs one red bead and plugs it to a blue bead, okay, somewhere in there. But as you can imagine, on the chromosome, if it does that, if it puts those beads together, there's now this big chunk of chromosome dangling down here, right between the two beads. So what happens is these nifty little molecular scissors, the RAG molecules, move in and they make a snip. They snip off that loop, and then the chromosome is glued back together. And it turns out that that loop is glued back together. And that loop in the T cell receptor, when it's made from the T cell receptor, this is again a loop of chromosome here that has been cut out. And it's got these conserved sequences where it was cut and glued back together. And so it turns out that you can use quantitative polymerase chain reaction, PCR, quantitative PCR, with primers to these conserved regions to actually quantitate how many of these, what we call T cell receptor excision circles, there are. Now, what you need to know about these TRECs, or these T cell receptor excision circles, is this. So they are made as the T cell is going through the thymus and being educated, okay? It's rearranging its T cell receptor gene, it's making these TRECs, okay? And because the each T cell only gets to do that once, it only makes so many TRECs. It just makes a few TRECs, okay? And so a newly, freshly minted T cell coming out of the thymus has several TRECs in it. And the beautiful thing about babies, you might remember, is that babies actually have lots of lymphocytes. They actually have about 3 times as many lymphocytes as an adult on average, okay? So babies have lots of lymphocytes, and all of those lymphocytes should be fairly freshly minted. They should be naive. And because of that, babies have really high TREC levels. They've got lots of TRECs, okay? So when you do this, when you measure the quantity of TRECs from the blood in a baby, there's many, many of them. Remember that as you get older, and you get to be an old guy like me, your thymus sort of involutes, and it doesn't work as well anymore, and your T cells are maturing, and as the T cells mature, as shown here, those TRECs, as this T cell divides and matures, the TREC goes with one of the daughter cells, but it doesn't go with both of them. So over time, those TRECs get diluted out as that T cell matures. So my TREC levels, adult TREC levels, are quite a bit lower than a baby, okay? Now, if a baby has SCID and has no T cells, there will, as a result, be no TRECs. And so when they measure that from the dried blood spot, they'll see no TRECs, and that signifies that a baby might be at risk for having SCID. And that's how babies get flagged, and we get the call as immunologists to do follow-up testing on those babies. So that's how this testing is done. You're looking for the presence of these naive T cells, using this by quantitating these T cell receptor excision circles. Okay, so that's how it's done. Now, it turns out that naive T cells, the beautiful thing about this assay is it's relatively easy to do. And it's very stable on the dried blood spot cards because it's a DNA-based test. And it turns out that all of the types of SCID are associated with low naive T cells. Even the leaky forms of SCID, like Omen syndrome, because those T cells just expand like crazy, they lose their TRECs. And so those patients, even though they might have some T cells, they don't have very many TRECs. And so this is an amazing assay. It really is incredibly sensitive for picking up babies who have low T cells, or by a surrogate of T cells, low TRECs, okay? So that's why it really works so terrifically. Now, this is really a new paradigm for detecting SCID. So remember that in the past, these are the boy in the plastic bubble, girl in the plastic bubble kids who would typically come to us, often at 3 or 4 months of age, after the antibodies from mom had sort of waned, they'd been metabolized, and the baby's now not able to make their own, and they would come to us severely infected, typically. And once they're infected, the chances, because particularly they have problems oftentimes with viral infections. We had one of the most recent babies we had before the start of newborn screening came to us with 5 viral infections and PCP pneumonia, okay, when he presented to us. And he did not survive because they're so infected that doing a transplant, it's very difficult to get them through. Now, the paradigm, at least in the state of Washington, is that we are typically notified by the Department of Health of the low TRECs on day of life 3, usually no later than day of life 3. Confirm it. We contact, the State Department of Health lab contacts the primary care physician who's taking care of the patient, and we have received, and that day typically the blood is drawn and sent for confirmatory testing, which is a flow cytometry test to look for the presence of T cells and B cells, etc. That is usually sent on day of life 3 or 4, and we have a confirmed diagnosis typically by day of life 5 to 6 in the state of Washington. These kids are totally well. They look fabulous, which raises one of the points that I will bring up a little bit later. Okay, we'll get back to that. We can stop breastfeeding so that there's no chance that CMV can be transmitted. If it's a CMV positive mother, we have them hold breastfeeding. No live viral vaccines so that we can catch them before they get their rotavirus vaccine, which is live viral. And if they are in the hospital and they need blood, make sure that they get irradiated blood products, okay? And we get them to bone marrow transplant before they get sick, and outcomes are dramatically better. So once you go through this, then you have to start looking for the genetic defect using the approaches that we've talked about. And as I said, there's now about 21 different genetic causes of SCID, and a growing number of those as we find sort of, again, the expanding phenotypes as we do this. Now, with regard to newborn screening, you can also do the same. You can detect both the TRECs from the T cells And you can do the same thing in the B cells, because remember, the B cells rearrange their immunoglobulin locus, in their immunoglobulin gene, and they form what we call CREX. So there's TREX and there's CREX. And if you combine together TREX and CREX, looking for both B and T cells, then you can detect even more diseases. So in this study published by Leonard Hammarström's group in Sweden, they were able to pick up a bunch of different forms of SCID and actually divide the ones that were B positive and B negative. They were able to pick up Nijmegen breakage syndrome, ataxia-telangiectasia, the X-linked A gamma globulinemia patients. So it's actually, so you get, by adding this one test, which you can actually multiplex right into that same assay, that same quantitative PCR assay, you can get a whole lot more diseases. And you can keep going. This is a platform that you can actually multiplex and do several tests all at the same time. So here's the group from Wisconsin who actually did a pilot study to look at whether or not they could detect DeGeorge syndrome using quantitative PCR for the TBX1 locus, which is in the middle of the 22q11 deletion. And it worked beautifully. It worked as well or better than the FISH assay. And it could be done for newborn screening, okay? And we've been working on a proteomic approach. You heard about proteomics earlier and the ability to use proteomics to look for metabolites. Well, we weren't looking for metabolites. We've chosen to look at marker proteins that are markers present on certain proteins. So for example, to detect SCID, we are looking for the presence of CD3, CD3 epsilon. For X-linked A gamma globulinemia, we are looking for the presence of BTK protein. For Wiskott-Aldrich syndrome, we're looking for the presence of WAS protein. And what we do is we take the dried blood spot, we elute off the proteins, we trypsinize them, we chew them up into little peptides, and when you shoot those peptides through a mass spec, you're able to get a tryptic peptide digest fingerprint of each of these proteins. And so we published this initial paper showing that we could detect successfully the difference between babies who had WAS, BTK, and SCID. Now the problem with this was that it wasn't sensitive enough to do on dried blood spots, but we've now added one more addition to this, which is to immunoaffinity purify the peptides before we do the mass spec, and in fact we can make this work on dried blood spots as well. And with mass spec, we could potentially detect 20 or 30 proteins simultaneously, and newborn screening labs also already have mass spec machines in their labs that they're doing the metabolic disease testing with already. So we think that there may be the ability to utilize this, okay? Now, what are some surprises that have come about as we've been doing newborn screening for SCID? Some of those surprises were really highlighted by this paper that was just published last month in JAMA, detailing the results of the total screening that has taken place since it began in 11 screening programs here in the U.S. And this is what came out of that paper. So first of all is that the SCID is more prevalent than we thought. So initially it was estimated that it was 1 in 100,000 births. We now know by screening that it's 1 in 58,000 approximately, which suggests that 1 out of every 2 patients with SCID was going undetected prior to the start of newborn screening. So there's a win already. We're picking the kids up. Second, it turns out that SCID newborn screening is a great screen for DeGeorge syndrome and for patients with congenital cardiac defects. In the state of Washington, overwhelmingly, the patients that have been detected by this assay are patients with congenital cardiac defects who also many times have thymic problems and have low T cells. So this assay is very good at picking up patients with low T cells. Doesn't necessarily mean they're SCID, but they've got low T cells. So the overwhelming majority of the patients that we've identified, probably out of around 20 patients, 18 of those have been either DeGeorge or severe cardiac defects, okay? And interestingly, I think this is an interesting point, getting back to the patients, getting back to the genomics piece, is that almost 20% of the patients with clear SCID, by all of the measures that we have, and pairing that with genomic testing, almost 20% still have no genetic diagnosis, okay? So there are still a number of things that we don't know that we need to figure out more. These are other things that have come up. Newborn screeners think differently than immunologists, and different, I would say, than many others. Newborn screeners worry about things like the fact that their task is to follow up on every baby that has an abnormal screen. That means they need staff. It is unbelievable. I wish we had newborn screeners in our clinic who could call our patients and make sure they're taking their meds, make sure they're showing up for their appointments. They are tenacious, and they follow up on every single baby. And that is their job. But that means that they need resources to do that. And so you can imagine this is resource intensive, and it requires them and that makes it so that when we come to them and say, oh, hey, let's throw in Trex with the Trex. We could get so many more diseases, and they go, whoa, let's not, because that means that we've got a whole bunch more false positives that we've got to follow up on, and a whole bunch more babies we've got to track. And they're happy to do it, but they are busy as well, and they are pushed to the limits of the resources. So that's one thing. The other thing that newborn screeners think about is what impact is that phone call that is received from the newborn screening lab going to have on a family and on that child. And this has become more, I would say, not problematic in maybe is in some cases, than I would have probably predicted up front. Because in the case of SCID, I think for some diseases you can say, your child has this, and we're going to put them on this special diet, and that's all fine and dandy. In the case of SCID, we are coming to families who have a beautiful, healthy, perfect baby. And we're sitting down with them and saying, your child has a fatal disease, and they are going to die if we don't give them a bone marrow transplant or gene therapy. And they're like, whoa, wait a minute. You got to be kidding me. I remember the last patient that we identified sitting in the room, breaking the bad news to the family, talking to the parents who were seated over here. Grandma's over here on this side holding the baby, and the parents are going along, and grandma's going, no way. He does not have SCID. No way. There's no possible way. And we'd go on a few more sentences. No way. He doesn't have SCID. There's no possible way. That's the boy in the plastic bubble. He is not the boy in the plastic bubble, which he wasn't. Okay? And we argued, and finally we got to the point, okay, how about if we send a second confirmatory test? Would that be okay? Yeah, that would be good. So we sent a second one, and she still was not convinced. So we sent a third confirmatory test, just to be sure that the first two were correct. And they were. And he had SCID. And so it took a lot of convincing. And I think that we've now come to realize that this, we've got to do better at this. And I think for any of you who may be having these patients in your practice, this is turning out to be a greater challenge than I think we had anticipated. And I think across the country, immunologists are having similar experiences. The third point is the question really of, now you've got a baby with SCID who looks great, they're not infected. How do you safely transplant a baby? Do you give them conditioning with chemotherapy and radiation and all of that stuff? And what does that do to a developing brain? Or do you just dump the cells in and hope that you get a good enough graft? I mean, we've got a fair bit of data to say that just dumping the cells in may not be sufficient. So how do we do this safely and not create long-term problems? That is an ongoing issue that I think we're going to have, that is being dealt with, but is going to take a lot more work. And lastly, it takes a community. It is unbelievable how many resources are marshaled for a single child with SCID. Unbelievable. The cost analysis has been done. The numbers, I think, are true. But when you're in the trenches and you're involved in it, it's mind-boggling to look at the work that goes into saving a single life. And it really, it speaks, I think, it's one of the things that to me it speaks to the humanity of what we do. And so those are things that have come up. So let me finish with just a few thoughts of where we're headed, or where I think we might be headed in the future with regard to early, rapid diagnosis of these diseases. And I'll start with this. This was a paper published in 2012 from the group at the Children's Mercy Hospital in Kansas. And they've got a very active genome sequencing core there. And they thought, you know, we have this problem that we have all of these NICU babies, and we don't know what they have. And wouldn't it be great if we could figure that out sooner and faster? So they went to work trying to figure out, you've heard how long it takes to get data back on whole exome sequencing. Some people said 3 to 6 months, right? So they said, not fast enough if you've got a baby in the NICU. It's got to be faster. So they dug in and started looking at where could they shorten the procedure to try and get to an answer faster. And they whittled their whole genome sequencing from blood sample to result down to 50 hours. That's an interpreted genome. Now, the interpretation is done by an algorithm. It's not by somebody sitting in front of it. So again, it's not perfect, but their idea was that if we could get an answer in 2.5 days for a baby sitting in the NICU, and even if we only got it for 25% of those babies, we could tell them this is, number one, we know what disease you have, and here's a therapy that might be useful. Or number two, we know what disease you have, and we know this is not compatible with long-term life. Let's start thinking about palliative care, making the baby comfortable so that we don't provide false hope for this child needing to survive. And the idea was that they thought they could pitch this, they could get it paid for because they would go to the insurance company. The cost of this, they still offer it, the 50-hour exome. The cost of this is $13,500. It's again, it's blood sample to results in 50 hours, okay? And they are pitching it to the insurance companies that $13,500 is less than a day in the NICU. So if you find an answer that decreases their NICU stay by 1 day, you've paid for the sequencing. And so that's the idea. That's where we are headed. That's where we are headed. The cost of sequencing right now, it's a race to the bottom, cost-wise. The companies are competing, falling over themselves to get it cheaper and cheaper and cheaper, and the speed faster and faster and faster. And that raises the question is this where we're headed? And I don't know if you have watched this movie, Gattaca. If you haven't, I would recommend it based on what you've heard at this meeting. There's a lot of food for thought in this movie. And it's really a society in which societal status is based on your genome. And it's absolutely fascinating. And I would recommend it. I don't think that's where we're headed, but it is a cautionary tale. And I think that it's a great sort of, rent it from Netflix. It's a great sort of Friday, Saturday evening relaxation project. All right. The last thing here is that, remember, we're only getting answers in 25% of patients that we do genome sequencing on. That means there's 75% that we're still not finding the answers on. And these are 75% of patients who clearly have a disease, very clear phenotypes. We've had experiences, we've had patients who are just unbelievably well characterized from an immunology standpoint, and we still cannot find the gene, despite our best efforts. So I think one of the things that's coming down the pike, and will come down the pike increasingly so, is beginning to combine whole exome, whole genome sequencing with whole transcriptome analysis. So basically beginning to look at all of those splice variants that I talked about earlier with that XLA patient. So looking at all of the mRNAs. Are they all there? Are the appropriate splice variants are there? Are they not? Are they missing? I know of one case in which that actually identified the defect. Combining the transcriptomics with whole exome sequencing. And then layering on top of that the immunophenotyping data that I showed you, and then proteomics. So basically taking mass spec, beginning to do proteomics to look at intracellular signaling pathways, but in a massive way, and layering together all of this data. And I think it's going to ultimately, to get at that next 75%, it's going to require this level of work. And the problem is, you've already heard about the data problem with a whole genome or a whole exome. Now imagine layering on top of that all of this other data. And the informatics problem becomes really crazy. Okay, so that's where I think that's where we're headed. I mean, there's already people beginning to do this, okay, and doing it on a clinical basis. I think that there will be expanded use of functional testing. You've heard about the use of mass spec, and it is going to grow. I think mass spec is a tool that we have just scratched the surface. And mass spec is going to be utilized in a much bigger way to look at in terms of diagnostics and trying to get at answers early. The beautiful thing about mass spec, again, is it is fast. It's fast, it generates a lot of data, and can screen, for instance, in signaling pathways in the cell, can screen hundreds of phosphoproteins to look at signaling pathways that are defective. And again, flow cytometry that I think we've begun to really mine, but there's a lot more that can be done. And then lastly, I think that there are new therapies coming down the pike that are going to be mind-boggling in what they're able to do. So first of all, we've got to get a diagnosis, and then we've got to have therapy. So we're getting better and better transplant regimens, less and less toxic conditioning regimens for transplant. And then, of course, gene therapy is just really sort of, we've been doing it for a while. There were a lot of initial problems with secondary malignancies and things, but I think we're getting past a lot of those to the point that gene therapy is really going to begin to accelerate, and it will not be long, probably less than 2 to 3 years, I would predict, before the first gene repair trials come about. And what I mean by gene repair is not actually taking a virus and putting in another copy of a normal gene, which is what you do in gene therapy to put in a good copy of the gene. What you do with gene repair is that you design a specific set of molecular scissors, basically an enzyme that goes in and finds that gene in the genome, makes a double-stranded break in that gene, and then you put in a template to basically replace that defective gene, but you replace it right at the site that it's supposed to be at. That is not far off, because we can make it work already in vitro in primary human cells. And it will not be, I would predict, more than 3 years before the first human gene repair trial is begun. So those are the things that I think we've got. What have we talked about? We've talked about the status of genetics in primary immunodeficiency diseases, the fact that it's growing. We're getting a new gene about once a month, once every 2 months, a new genetic defect. We talked about how genetic defects are identified in immunodeficiency diseases and some of the surprises that have come up as we've been doing that. We've talked about the importance of using functional confirmation of genetic defects that are found and having functional tests available at hand and ready so that once you get the genetics, you can make sense of it. We've talked about newborn screening for immunodeficiencies, how that's being done, and some potentials there and some surprises that have come up related to screening. And we've talked a little bit about where I, at least, feel like we're headed in the not-so-distant future. So with that, thank you very much for inviting me and for listening so attentively.